Part 3 of Reproductive Endocrinology. We're going to go over the menstrual cycle. This is going to be the last of female reproductive endocrinology. Next will be male reproductive endocrinology. Now the monthly cycle, everyone will say the monthly cycle is 28 days. That's true, but it's also 28 days plus or minus 7 days. So 28 days is key. That's what everyone's going to remember. Just remember that the average can be 28 plus or minus 7 days. So if it's easier for you to remember 21 to 35, then remember that. The actual length, so in the female monthly cycle, you have a couple of phases. So you have the follicular phase, and you have the luteal phase, and then you have, of course, menstruation being at the beginning of the follicular phase, the early follicular phase. So the variable part of the cycle is typically the follicular phase. If you have a difference in the 28-day cycle, it's probably a difference in your follicular phase. The luteal phase is pretty constant among all women. Usually at 14 days, it can be at 13 days at times, but the average is 14, so just remember that. The average time of menstruation is 4 days plus or minus 2. And the average menstrual volume is 20 to 60 milliliters. Or if we want to keep with our plus or minus thing, we can say 40 plus or minus 20 milliliters. So right down here I have written regular irregulars. So what I mean by that is there are certain times when it is normal to have an irregular cycle. So when is it normal to have an irregular cycle? At menarche, meaning your first period, for the 2 years following that, the cycle is usually very, very irregular. And then if you take 3 years prior to menopause, cycle is very, very irregular during this period as well. The take home to this being, if you're late in life and you're noticing your cycle changing, then it could be an indication that that menopause is within 3 years from where you're at. A person's cycle is most regular between the ages of 20 and 40. So the most regular. So if you have an irregular cycle between these ages, that's extremely irregular. So from now on, we're just going to call the 28-day cycle the normal cycle. Although normal has a range for between 21 and 35, we're going to call 28 days the normal cycle. Now, of course, I used a couple different texts and some research papers when I put this together. The first text being Costanza's Physiology. She says that the first day of menstruation is day 0 of the cycle, whereas Williams Gynecology calls it day 1 of the cycle. Personally, I prefer this nomenclature just because calling something a day 0 is kind of an oxymoron. For example, a baby is born. They're not 1 year old yet. They are 0 years old, but that is their first year of life. So you don't call that the 0 year of life, you call that the first year of life. So in a standard 28-day cycle, you're going to have basically 3 phases, or you can break it down into 2. You can break it down into the follicular and the luteal, which most texts do, or as Williams does, he breaks it down into 3. So you have the early follicular, you have the follicular. So these are all really the same thing, but what we're doing is we're breaking down these endometrial phases. So the endometrial phase, the menstrual phase, it corresponds to the early follicular phase. And then during the rest of the follicular phase, the endometrium is proliferating, it's getting larger, it's growing, it's under the influences of estrogen. Estrogen causes the endometrium to grow, so we call this the proliferative phase. So proliferative phase of endometrium and follicular phase of the ovary are the same thing. Then at this point right here, you get ovulation. And from ovulation forward, the ovaries are what's called the luteal phase, and under the influence of progesterone, the endometrium starts to become secretory, so it's the secretory phase. So over here, just to note, I have this written as E2 and P4. E2, just to note, is estradiol, and P4 is progesterone. The reason for the numbering nomenclature is because there's actually an E1 and an E3, E1 being estrone and E3 being estriol. These are all three different types of estrogens, so you have to have a numbering system. And like I said, the ovulation marks both the end of the follicular phase and the beginning of the luteal phase. And the variability mostly comes from the follicular phase. So if you vary, like I said, from a 28-day cycle, it's because your follicular phase is either shortened or lengthened. So the ovaries are kind of the key players. They're the intermediate player in the monthly cycle. So you have the hypothalamus and pituitary, which we talked about in the last video, and then you have what they do to the ovaries, and then the ovaries basically banking and doing all the rest of the work in the body. So where do the ovaries come from? How do they develop? Well, you got three basic cell types. Of course, there's more cell types, but the main cell types, you have the germ cells, which are called oogonia, and so they're called oogonia when they're surrounded by the granulosa cells. They're called oocytes. They're coming from the endoderm of the yolk sac. So in early development, that's where they come from. Granulosa cells, they come from the coelomic epithelial cells, and the ovarian stroma, they're the mesenchymal cells of the gonadal ridge. Now with that said, I don't think this is particularly high yield as far as medical school goes. For research, this is probably high yield if you're doing MD, PhD. However, these timelines, a little bit more important in my mind. So what happens, week 3, you get the primordial germ cells becoming present. So you get the first of these primordial oogonia. By week 8, all of the enzymes needed for the production of estradiol, estrone, estriol, all of the enzymes become present, but they're not really used. Even as estriol is produced during pregnancy, it's not being produced by the baby, by the fetus. It's being produced by a combination of the mother, the placenta, and the fetus. And the fetus is only making the androgens. The placenta is actually turning the androgens into estrogen. But the enzymes for that, they are present at 8 weeks of gestation. Between 10 and 12 weeks, the ovaries and the testes become histologically distinct. So if you were to take out the gonads of a fetus prior to the 10th week, you would not be able to determine whether those gonads were testicles or ovaries just by looking at them under a microscope. At week 12, the primordial germ cells begin to enter into meiosis 1. So several of the primordial germ cells are undergoing something called mitosis to increase their number. But then, around week 12, some of them slowly, one by one, enter into meiosis 1, and that's when they become a primary oocyte. By week 20, through mitosis, the maximum number of oogonia is reached, and that's between 6 and 7 million. I think most texts will say 7 million. Some will refer to 6 to 7 million. I don't think anyone has the ability to count these, so a rough estimate's great. All right, now, first thing I want to say is, so oocyte development. The first thing is the stage, these numbers, these are just numbers that I made up and put on a list of things as far as the development of the oocytes. The numbers are not important. They're just there to keep your mind straight. Now the second thing, what is actually important, is we see that right here we have this follicular development. The primordial follicle is arrested at prophase 1. Well, I just said that the primary oogonia or oocyte is arrested in meiosis 1 or arrested at prophase 1. So the point that I'm making is that the primary oocyte and the primordial follicle are the same thing. So from the primordial follicle, you can get a primary follicle. Now, the thing I want you to know about these, the development from primordial follicle to primary follicle, this is called gonadotropin independent. So gonadotropin independent. That means it doesn't require any FSH or any LH to make this transition happen. And it may even be that transition between the primary follicle to the secondary follicle is also gonadotropin independent. And what's being discovered kind of on the edge of the unknown is that the transition between gonadotropin independent and gonadotropin dependent is sort of a gray line, and no one knows exactly when one stops and the other one starts. So with that said, from secondary to tertiary, this is gonadotropin dependent. So the secondary follicle, once you get to that, it's called preantral. It's called preantral because there's something in the follicle called an antrum whenever it gets to the tertiary stage. The tertiary, Graafian, and antral follicle are all the same word meaning the same thing. And anyhow, in this tertiary follicle, you have if you look at it under a microscope, it looks almost like a little lake of fluid that gets built up in among the granulosa cells, and we're going to take a look at a picture of that in a minute. But in the secondary follicle, that antrum either hasn't quite formed yet or it's beginning to barely form. The main thing about the tertiary stage, besides that there's an antrum there and that antrum sort of concentrates some of the hormones around the oocyte, the thing about the Graafian tertiary antral follicle is that it finishes up meiosis 1. So it finishes up meiosis 1, and when it does that, instead of arresting at prophase 2, it arrests at metaphase 2. Metaphase 2. So what causes this arrest of meiosis? It's something called oocyte maturation inhibitor. It's abbreviated OMI. And exactly the cell signaling that turns on and off OMI, I'm not certain of. I was unable to find, so I don't know if that answer is known or not. But I have sort of a good idea or a good working hypothesis based on all the other reading and things that were said about various hormones. Nothing specifically said this turns on and off OMI, but different hormones induce the finishing up of meiosis. So once you're arrested at metaphase 2, you go into ovulation, and so whenever the oocyte is ovulated, it's called a pronucleus. It is at that point, it's spitting out this polar body, so it's a haploid cell. And so what the polar body is, if you know anything about biology, which I'm assuming you do, you know that whenever you undergo meiosis, you usually will split the DNA in half. However, with an oocyte, it splits the DNA in half, but the half it wants to keep, it keeps in this nucleus, and then the half it wants to get rid of, it kind of pushes over towards the side, and then it kind of gives it the least amount of cytoplasm that it can and gets rid of it. It expels it. So the oocyte is trying to preserve its cytoplasm, preserve all of its organelles. And so to do that, it creates this polar body with the extra DNA. Instead of making 2 germ cells or 4 germ cells, it makes 1 germ cell that's ready for ovulation out of the 2 phases of meiosis. Whereas in sperm, you're going to get 4 sperm out of 1 germ cell. Whenever fertilization occurs, it's then there's several names for it. One would be pre-embryo, another one would be zygote. So another one, you just say baby if you want. Now the last point I want to make is that all the way up until ovulation, the follicle is avascular. It isn't until ovulation that you get vascularization of the corpus luteum. So what that means is that there are lots of cell junctions, and the egg relies on diffusion for its nutrients. Now, just to be clear, meiosis 1 begins right here in the primordial follicle, and it stops at prophase 1. Once you get to the tertiary follicle, prophase 1 goes on and all of meiosis 1 gets completed. At that point, it arrests itself at metaphase 2. In this metaphase 2 state, it gets ovulated, and it's still stuck in metaphase 2, and it doesn't finish up the second meiosis unless it gets fertilized by a sperm. So when sperm fertilizes the egg, it quickly finishes up meiosis 2. So here's a cartoon I was able to get off of OpenStax. OpenStax is kind of a free source for information. You can just download these. And what they have is a great cartoon of the follicular development. And so right here we have our primordial follicle. And the thing that I want you to notice about the primordial follicle is that yes, right here it is surrounded by granulosa cells, but the granulosa are flattened. And you don't see any theca cells at this point. Then in the primary follicle, you'll see that the granulosa cells become cuboidal, and you get something called a zona pellucida. Basically what happens is we're secreting, the oocyte's secreting proteins, and those proteins form something called the zona pellucida that protects the oocyte. The proteins, we've named them ZP1, ZP2, and ZP3. So ZP standing for zona pellucida, and the number being the number of the protein. Now it's thought that ZP3 functions in basically guiding sperm, saying, hey, this is who I am, come and eat me. And ZP2 is a hardening factor, so whenever the sperm opens its acrosome, whenever the first sperm gets in, the acrosomal head busts open, it releases an enzyme that hardens ZP2 and prevents other sperm from fertilizing the egg. Now you know you're at a secondary follicle when the surrounding stroma cells start differentiating into theca cells. So the theca cells will start differentiating from their surrounding stroma cells, and it will surround the granulosa cells, and they'll produce a basement membrane basically that separates the two of them. At a late secondary follicle, you start to see the production of the antrum. So then, of course, the tertiary follicle is noted because of the large antrum that it has, and then also you get two types of granulosa cells. So the granulosa cells that surround the oocyte are called cumulus oophorus, and so these are the granulosa cells around the oocyte, and then the granulosa cells around the antrum are called mural granulosa. And so I'm sorry, I'm not able to spell very small. I'll write it bigger. Mural granulosa. So then, of course, you have ovulation, which just takes a few hours, and then the cells begin to transform into the corpus luteum. This transformation is one of the greatest examples of cell differentiation that happens in adult mammals. So again, I have this image from OpenStax, and you see the oocyte. This is a secondary follicle oocyte with the zona pellucida around it. You can see some of the thecal cells out here on the ridge, so I'm highlighting with red. Of course, the granulosa cells are quite easily distinguished, and then you start to see this developing antrum right here. So this is about to become a Graafian follicle. And of course, I've relisted those stages, and I've put some notes about what you'll see in each of those: the flattened granulosa cells, cuboidal, many gap junctions, zona pellucida, the proteins talked about, and then also down here, the antral fluid. It contains high concentrations of hormones and secretions, so it's going to basically concentrate estrogen higher in that area than what it would be in the rest of the body. So you basically feed forward on all of the stuff that estrogen does, such as upregulating LH receptors and that sort of thing. It's also going to concentrate other growth factors, for example, IGF. So the timeline. Okay, so what happens? How do we finally get our ovulation, which is basically the goal of all this? 3 months before any thing ovulates, you have the recruitment of a cohort. So cohort recruitment. So basically, it's saying we're going to take a certain number of oocytes that we have in our reserve pool, and we're going to tell them to turn on a certain number. So usually that is 3 to 11 oocytes for each ovary, which would mean 6 to 22 oocytes altogether. And there's a wide range, and that range is basically it's correlates with the number in the inactive pool. So if you're really young, you have a large inactive pool of oocytes. So for example, whenever you start puberty, it's something around 400,000 oocytes are available, and by the time you're about to reach menopause, that number has declined drastically. So the number that are recruited each month corresponds or correlates to the number in the inactive pool. So let's go with the big number, 22. We selected 22 this month, and in 3 months, one of those 22 will win the race and become the ovulating oocyte. What happens to the rest of them? Well, they become atretic, which means basically they program themselves to die based on certain signals. We'll take a look at a few of those signals here in a moment. Now, during the last month, there's this concept called the selection window. And the selection window is basically one of the oocytes has to become dominant. So, am I the dominant follicle? Well, you won. And that's tightly controlled by FSH. So what happens is basically, if you have, there was a recent study done I was looking at, if you add FSH to a person throughout the entire month, give them excess FSH, you get ovulation of multiple oocytes, which can mean, twinning. For example, you can fertilize all of those oocytes as well and get multiple babies at delivery. So as the estrogen levels go up, and so estrogen and inhibin B during the follicular phase, they are going to feed back on FSH, causing it to decrease. And by decreasing, any oocyte that is not 100% ready, it's going to stop receiving that FSH stimulation, and then it's going to say, I wasn't ready, FSH is gone, I'm going to go through apoptosis now. And that's sort of the halfway understanding of it, and nobody knows all of the ins and outs of it just yet. So I say it's controlled by FSH and really a decrease in FSH, but there's also probably some other fine-tuning factors as well. So let's talk really quick about atresia. I said that some of the oocytes, the ones that don't become primary, they become atretic, they go through apoptosis. Well, it's not just the follicles that were recruited and didn't make it. You start at 20 weeks of gestation with 7 million primary oocytes. By the time you go through menopause, you ovulate about 500. So some of this is because of the selection window we just talked about. It's either you become primary or you die. Some of it is because there is no, after that 20th week of gestation, there is no more production of germ cells. So no more germ cells, or basically no more germ cell mitosis. And what that means is like with sperm, you have this supportive germ cell layer that is constantly undergoing mitosis to produce more and more and more sperm, and then those products undergo meiosis later on. But you always have this reserve layer of germ cells that can undergo mitosis several times to produce more and more and more. This doesn't happen with females. They get what they have when they're born. But that still won't account for this loss of 6,999,500. It'll account for a small amount of that, so where does the rest of it go? It's probably tightly controlled, and no one knows exactly for sure, but every month more and more of these 7 million cells will undergo apoptosis. It's not necrosis. It is not some type of avascular or vascular necrosis. It is a programmed event, apoptosis. We just don't know all the programming yet. So by the time you're born, you go from 7 million to, this is supposed to say 1 to 2 million, not 100. So just scribble out the zeros. So 1 to 2 million at birth, and then by the time you hit puberty, you have 400,000. So this is before any cohorts have been selected or anything. You've lost that many oocytes, and then you'll go on to ovulate only about 500 of those. And then, as I've said, it's controlled, it's not necrosis. The main idea of why this happens, the purpose for all of it, is you want to select the most fit oocyte. Why do you want the most fit one? Because you want to make superhumans. You want to be able to make something that's going to survive, essentially. You might not look like me right here in this picture. That's me. But you want to make the strongest thing you genetically can make. So as far as selecting the dominant follicle, like I said before, it's sort of a race. Whoever can make it to producing the most inhibin and estrogen first wins. However, there's some other characteristics of a dominant follicle. For example, there's high IGF levels. There's also high estrogen levels. IGF and estrogen have been shown to prevent apoptosis. There's high FSH concentrations in the antrum, and so it's able to concentrate the FSH and other hormones. There's also high FSH receptor expression on these follicles compared to the follicles that don't make it. And then, interestingly, there's high VEGF production around the follicle. So we said that the follicles up until ovulation, they are avascular, but they have some. So if this is my follicle, I can have, blood supply going around it. It just can't have anything going through it until after ovulation. And so what does VEGF do? VEGF stands for vascular endothelial growth factor, and so it causes these arterioles and arteries and capillaries to grow more and more and to basically give it more blood supply. If it has more blood supply, it has more exposure to the hormones. It's able to concentrate the hormones more and faster as well. So here we have a picture of the ovarian histology, and basically the three main layers. You've got the cortex, the medulla, and the hilum. And I want to reiterate, because as you read about these kind of things, you're going to see over and over again referring to granulosa cells and theca cells. Granulosa cells are going to surround every primordial follicle. You're going to have anywhere between a thin flattened layer all the way up to the massive cuboidal layers, pseudostratified layers, and so on. But what about the theca cells? The stroma cells surrounding a follicle differentiate into theca cells. That's the magic. So what is important about each layer? So the cortex out here, we're going to have the follicles and germinal epithelia. In the medulla, you have connective tissue, of course. Then you have these myoid-like cells. Myoid-like cells are basically cells that act like muscles. They contract. And, of course, the interstitium. The hilum is going to provide the vasculature for the whole thing. It's going to have lymph nodes. It's going to have nerves. And it's going to have interstitial cells that resemble Leydig cells in the male gonads. The function of these special interstitial cells are not exactly known. However, their proximity to the vasculature sort of indicates that they might be a micro-signaling cell, letting the other cells know exactly what's going on in the body. Let me clarify that. They are letting the ovarian cells know what's going on in the rest of the body. Now, because they resemble the Leydig cells, so these interstitial cells in the hilum, because they resemble the Leydig cells, if they become if there's a hyperplasia or a neoplasia forming a tumor of these specific cells, you can get some testosterone and androgen secretion. And so if you have a high androgen testosterone level specifically, you might check for a tumor in the hilum of the ovaries. So one of the main functions of the ovaries is to produce estrogen. And so there's something called the two-cell theory on that. How is the estrogen produced? This was a theory proposed back in the 1950s. It's pretty well established. So what you have is we're going to call this the granulosa cell. Then you have this basement membrane, and you have the theca cell. The granulosa cell is going to have a receptor on it, a receptor for FSH. So So let me just zoom in here so I can write FSH. So the granulosa cells have FSH. The theca cells have receptor for LH. So what's going to happen is we're going to have cholesterol in the blood. So this is my blood. And actually, let me make this red. So this is my blood stream. Cholesterol from the blood is going to get transported into the theca cell. And under LH influence, it's going to activate cyclic AMP. And cyclic AMP, through transduction, is going to activate all of the enzymes to turn the cholesterol into an androgen. One of many androgens. That androgen will then seep across the basement membrane. I say seep, biological term is diffuse. Under the influence of FSH, we're going to get activation of cyclic AMP, and it will activate all of the enzymes, especially aromatase. And aromatase will turn the androgen into estradiol, or E2. The E2 will then go back out into the blood. So that's where estrogen in the entire body is coming from for the most part. You'll have a little bit of peripheral conversion of various androgens in the adipose tissue and in the skin to estrogen, but for the most part it's all coming from these granulosa cells. Now it's important to note the rate limiting step for converting cholesterol into androgen is processed by CYP17. CYP17 is expressed in the theca cell, but not in the granulosa cell. And then equally important, aromatase is expressed in the granulosa cell, but not in the theca cell. So in order for cholesterol to be converted into estrogen, it has to first be processed by the theca cell, transferred into the granulosa cell, and then converted into estradiol. So in the ovary, the major secretions are estrogen and progesterone, and the minor secretions would be estrone, anastradiol, testosterone, 17-alpha hydroxyprogesterone. So these are very minor and have less influence than these major secretions. Progesterone, however, can be totally taken care of by the theca cells. All of the enzymes are present in the theca cells alone to convert the cholesterol into progesterone. However, this doesn't start occurring until the very end of the follicular stage, and it reaches a maximum in the luteal stage. So hormones are being produced all throughout life. At 8 weeks of gestation, you have all the enzymes for production of estrogen. However, the baby itself is not actually producing any estrogen. As you can see if you study the hormones of pregnancy, which I'm not going to cover, the estrogen is produced by a combination of the mother providing cholesterol, the placenta converting that into various products, the baby converting that into various other products, and then the placenta acting on the final product to produce estriol. However, the baby itself at 8 weeks has all the enzymes to produce estradiol. The baby's production of FSH and LH is actually going on, and it will remain high until into the second trimester. That's when it's believed that the hypothalamus becomes sensitive to the placental produced estrogen. So from that point on, FSH and LH levels will drop until birth. At birth, and this is my little boy, Justice. So at birth, the baby is freed from the inhibition of the placental hormones. At that point, FSH and LH levels go back up, and they stay up for approximately 2 months. Then, at 2 months old, the central nervous system starts inhibiting the hypothalamus. Now, exactly what's going on is still not known. For a long time, it was thought that there was enhanced negative feedback. So what that means is basically that a little baby will have a super small amount of estrogen being produced. And so that they thought at about 2 months of age, the hypothalamus goes into this stage of enhanced negative feedback where even a little bit of estrogen will inhibit its activity, keeping it from producing LH and FSH. However, studies with gonadal dysgenesis, where you have no hormone being produced at all, the LH and FSH levels are still low, and so it's thought that it's the CNS developing to the point that it just causes the inhibition from other areas of the brain. Now, see, we talked about babies, and then the next thing that happens is there's puberty. So let's talk about LH and FSH levels. At premenarche, the FSH levels are greater than the LH levels. And then at puberty and we'll just say reproductive age, you have LH levels going higher than FSH levels. Now, these are relative to the period in the cycle, but just as a general rule, this is what happens. And then at menopause, you have the reverse going to FSH again. FSH levels greater than LH. So premenarche and menopause have high FSH levels, whereas reproductive age has high LH levels. Then if we just take that and look only at the LH levels, so let's say that on our y-axis we have LH levels. Then on our x-axis, we're going to break it up into a couple different things. So we're going to put childhood, puberty, reproductive years, and menopause. Okay, so now we're going to do one more thing where we break this up. We're going to have day and night, day and night, day, night, day, night. So we're going to look at how there's changes between the day and the night in each of these stages of life. So during childhood, LH levels are basically pretty low. Then once puberty hits, they stay low in the daytime, and then at nighttime, they jump up. Reproductive years LH levels stay fairly constant through the day and the night, and then they jump up again and stay constant in the menopause years as well. The reason it jumps up right here in menopause years is because typically you would have negative feedback of estrogen hitting the hypothalamus, but now you have no estrogen being produced in the ovaries, so you don't have any negative feedback, so you just maintain that constant output from the hypothalamus and pituitary at a high level. But what's really interesting about this whole thing is that if we isolated just this area right here for puberty, let's isolate this area right here in puberty, right? This is before menarche, and what you get is that this fluctuation between LH levels produced in the day and in the night fluctuate up and down. And this can be one of the indicators of puberty going on, besides the outward changes. This would also be something that you could evaluate if you're looking at various other endocrinology problems in reproduction. So you want to say, okay, have they went through puberty yet? Are they going through puberty? You would measure daytime, nighttime LH levels to see what they were. So now let's put this with what we just learned about FSH and LH levels together. So we found out that they stay low right prior to birth, and then at birth they jump up for about 2 months. This is going to be my FSH levels. And then it jumps back down to a low base level up until, and stays at this low base level up until puberty. At puberty, it jumps up again and it starts fluctuating. It does this fluctuation all throughout the reproductive years, and then at menopause, it jumps up. So this would be FSH. Then LH levels, of course, is going to be going up and down with it, but during childhood, they're going to be lower than FSH levels. Right after puberty, it's going to jump up and be above FSH levels, having the same cyclicity, and then during menopause, it will drop back down below FSH levels. Now I want to point out before I move on to something else that this cyclicity that I drew in right here is just the normal monthly cycle. This is not representing the day and night cycle seen right at puberty and before menarche. Because frankly, that would have all taken place like right at one point on this graph, and the graph isn't big enough to show the day-night cycles for pubertal young ladies. So what we can see though here is that the gonadotropins go up at puberty, and increased gonadotropins equals increased estrogen. So what effect does estradiol have? You get a growth spurt, you get thelarchy, you also get adrenarche, you get pubarchy, menarche, and all of this happens over a period of 3 to 4 years. And let me talk about what each of these things mean. The suffix arche or arche comes from the Greek alpha, rho, chi, epsilon. And this just simply means beginning or first. So when we're talking about thelarchy, that's the beginning of the breast development. Adrenarche, that's the beginning of when the adrenal glands start changing their production mode. Then you have pubarchy, that's the beginning of puberty. Menarche, that's the first menstrual period. So let's talk about a little bit of the fluctuations of the menstrual cycle. Whenever you have FSH stimulation, it increases estrogen and it increases inhibin B. Both of these things feed back and inhibit the production of FSH. So the question becomes, where does inhibin come from? Well, there's a lot of evidence that we know that it's produced in the ovary, but is that the only place that it's produced? And the answer to that seems to be yes. For example, if you have a surgical removal of the ovary, inhibin gone. However, follistatin, one of the other hormones we talked about in the previous video, it does not change. It stays constant just like it was in the past. It has no change. Follistatin actually does not change with the cycle either, so there's no cyclicity. And the ovary is famous for its cyclicity, so it indicates that it's also it's definitely not the ovary producing follistatin. So in addition to stimulating estrogen production, FSH causes proliferation of granulosa cells. Proliferation of the granulosa. And the more granulosa cells that you have, the more estrogen you can produce. So you get a surge of estrogen late follicular phase. You get this estrogen surge. So this whole time estrogen levels are going up, they surge. This whole time FSH levels, on the other hand, are going down. And then at this surge, something changes it from negative feedback to positive feedback. No one knows exactly why. What is known is that if you have greater than 200 picograms per milliliter of estrogen for longer than 50 hours, that will cause a switch from negative feedback to positive feedback, and it causes the FSH level, the FSH levels to surge. This is where you have ovulation, and switching from follicular phase to luteal phase. Now, we also talked in the last video how at a high estrogen level changes the hypothalamic pulsatility, the rate, causing it to favor LH, over FSH. So that's what's about to happen. We're going to have an increase in LH production. So we had LH down here, and of course it spiked with FSH, but it went above FSH. So as it comes back down, it's still going to stay higher than FSH for the rest of the cycle. So we had FSH spike, come back down, and they will both continue to fall for the rest of the cycle. However, until the end of the luteal phase, LH will stay higher. Now, LH favors the production of inhibin A. So there's still some inhibin B being produced and causing a negative feedback on FSH. Inhibin A is also being produced to, I guess, augment that now that the estrogen is declining and the progesterone is beginning. Now, like I said a minute ago, follistatin and activin stay constant throughout the cycle. So I just said this, but I'll repeat it for your memory. FSH leads to increased inhibin B, which leads to decreased FSH. An important point to remind you is that IGF is really important for fine-tuning the cycle, and FSH leads to an increase in IGF binding proteins. So it's basically a functional reduction in IGF. So the quicker we get FSH out of there, the quicker we get rid of these binding proteins, and IGF can go in and take its role in the fine-tuning of the ovarian cycle. Now, of IGF, IGF-2 seems to be the most important. Under LH production, inhibin A becomes more dominant and inhibin B peaks and then begins to fall. So what I want to do is see if I can't draw out this classic graph that we always see when the menstrual cycle is being discussed. So imagine, if you would, that we have labeled 1 through 28, and right here is the separation between the follicular phase and the luteal phase at day 14. So this is day 1, 14, 28. So first let's talk about FSH, the gonadotropin. So FSH levels are starting to rise from the end of the previous cycle. At the end of the previous cycle, FSH levels rise and it starts the selection process of the cohort. And so they continue to rise and fluctuate up and down. And then because they're causing estrogen to be produced, around toward the end of the follicular phase, the FSH levels will start to drop. So let me pause there. I'm just going to write down here in red, this is FSH, and now I'm going to switch and talk about estrogen. So let me just put a little line here. So this is sort of a new baseline. Everything above this line is talking about the ovarian hormones, estrogen, progesterone. So I have estrogen, and it's slowly rising throughout the follicular phase until it gets to about here, and it starts to go up quite rapidly, reaching a peak and then coming back down right here. So what happened? What happened was we had the FSH causing the production, so the FSH causing the hyperplasia, the production of more and more granulosa cells, and that allowed for an increase in estrogen production. The estrogen being high initially causes the FSH to go down, but then when estrogen is at a level of 200 picograms per milliliter for longer than 50 hours, it switches everything and you get a spike in FSH. Now, something else that estrogen does is it starts to increase the number of LH receptors on the theca cells. So you have LH, it's dropping. Let me actually change this color to blue. So it's dropping from the previous cycle, and then it stays pretty low throughout the beginning of the cycle. However, we know that we have some LH being produced and some FSH being produced because without both being produced, the two-cell theory for the production of estrogen wouldn't work. So in order for estrogen to be produced, some LH and some FSH has to be being produced constantly. So a high amount of estrogen, remember, it's going to change the cyclicity of the hypothalamus, causing it to begin to favor LH. So right down here I'm going to put LH in blue so we remember that. Also I'm going to put in green E2 for estradiol. So that increased estrogen changes the cyclicity and we get an increase in LH, and then also it will surge as well, and it will surge much higher than FSH. Now what about progesterone? I'm going to put down here in a yellow P4 for progesterone, and we're going to go ahead and try to mark this off. Progesterone will be above this line. I won't be able to do it quite as high as I would like to, but I think you'll get the point that you have almost no progesterone production. It's literally almost zero for most of the follicular phase. And then at about the peak of the estrogen, progesterone starts to go up. So this is the one part of the cycle I don't completely understand, except that maybe you have a change from granulosa cells and theca cells to something called theca lutein cells, and maybe that differentiation is taking place at perhaps a certain level of estrogen or a certain level of LH. I'm not sure which. So why this begins to go up right here, I'm not exactly sure. It's the one part of the cycle that's still confusing me. And the reason I say it's confusing me is because I found in no textbook and no publication that says we don't know why this happens. However, there's never an explanation for why it happens. So pop quiz time. What happens now? We have ovulation. At this point, ovulation separates the follicular phase from the luteal phase. Exactly how ovulation happens is something else the textbooks will say we don't know exactly, but what we do know is some of the chemicals involved. For example, epidermal growth factor like family, plasmin, TPA, collagenase, prostaglandins. All of these things are involved. For example, prostaglandins, a woman that's undergoing fertility treatment, she will often be advised to avoid prostaglandin synthase inhibitors because whenever the prostaglandin levels are too low, you might not get ovulation. So anyhow, back to what I don't know. I don't know why the progesterone level goes up right here. What I do know is that this increase in progesterone sort of augments the estrogen in all of the changes, the feed forward, the positive feedback mechanism that switches on here. And the reason we know that is because whenever you, one sort of anecdotally, if you inject a woman with progesterone during sort of the late follicular phase, if she's anovulatory normally, it can cause an ovulation. So now we've had this LH surge, and what that does, it's going to turn off the genes of the follicular phase and turn on the genes of the luteal phase. That's what the LH surge, its primary thing that it does. Now during this whole thing, inhibin B is going up and inhibin A is beginning to be produced, and so these two inhibins may be may possibly be the mechanism that turns off this surge. Regardless of what turns it off, what's going to happen is it's going to drop precipitously back down, and then it's going to kind of level off and taper for the rest of the cycle until it gets back down to where it was at the previous cycle. FSH also will begin to drop down, and it will stay under the LH levels until the very end of the luteal phase, where it jumps back up, not as high as what I'm showing here, but it jumps back up like I showed over here to start the recruitment of the next cohort of follicles. Estrogen levels, after it reaches that spike, they continue to fall rapidly at first and then slowing down, and then toward the end of the cycle they fall back down, allowing the LH surge. So there's no longer any negative feedback on LH, so it allows the LH surge, and during that LH surge it causes the estrogen to start going back up for that next cycle. Progesterone, on the other hand, still a slightly different story. So it continues to grow until it gets about halfway through the cycle, the luteal phase of the cycle, and then it falls back down to about next to nothing again for the next cycle. Now you'll notice somewhere right around the LH surge, the progesterone does appear to be going up. However, and I drew this fairly accurately actually, when you compare it with, for example, Linda Costanzo's graph, it's way into this LH surge before any progesterone starts being produced. It's being produced primarily by the theca cells, and we know that the theca cells are already expressing LH receptors because the production of estrogen requires it. So why isn't the LH causing progesterone to be produced until a certain level, or maybe until the estrogen peaks out? I don't know. However, from that point on, progesterone, so the theca cells are all recruited, so they're constantly recruited during the follicular development, and they're getting pumped up into overdrive, so they're ready to go right here, but then as the LH levels start to wane, the progesterone levels start to wane as well. Now what about the endometrium? Well, estrogen's going up, causing the endometrium to build up its layers, and then progesterone causes the endometrium to be sustained and to start secreting various things. So building up, sustained, and secreting. And then whenever the estrogen and the progesterone are all back down to nothing, there's nothing either sustaining it or building it up, and so you start to get necrosis. So that's how you get menses. However, before we talk about the uterus and endometrium specifically, let's cover a few more changes that happen. So we know this follicular phase fairly well, and this luteal phase we still need to know a few more things about. So the first thing we need to know is that the basement membrane disintegrates gone. This allows for vascularization, and then you have blood supply going to both the theca lutein cells and the granulosa lutein cells. So they've changed into these lutein-like cells, lutein meaning yellow. So this is the corpus luteum, which just means yellow body. Now whereas the follicular cells were primed to produce estrogen, the corpus luteum is primed to produce progesterone. It's like it changed its specialty. Now, if there's no pregnancy, you get something called luteolysis, which is basically this becomes fibrotic, and apoptotic. I'm just going to write apoptosis. Whenever this happens, at first the product of this is called the corpus albicans. Albicans, you may think of albino, for example. Albicans means white, so this is the white body, white because of the fibrosis that's going on. Now, if there is fertilization, you get HCG rescue, which means because the LH levels are going down, in order to keep the corpus luteum going, it needs to have things interacting with the LH receptors. HCG, being very homologous to LH, will bind to the LH receptors and keep the corpus luteum going and producing progesterone. Why is it important that progesterone be produced? Only progesterone can sustain pregnancy. It can sustain the endometrium. I hate it when I'm writing and talking because I invariably always make spelling errors. Now this is important because the corpus luteum is producing all the progesterone for pregnancy up until about the 10th week, so around the second trimester. That's when the placenta, you have a placental, luteal placental shift, I think is the correct term, luteal placental shift. And so if you have, for example, some need, for example, a tumor, if you have a tumor in the ovary and you need to remove the ovary, if that is the ovary that contains the corpus luteum, you're going to have to do one of two things. You're going to have to wait until week 10, or you're going to have to administer progesterone. It cannot be a synthetic progesterone. It has to be actual progesterone, because only true progesterone can sustain pregnancy. So here we have basically a picture of the ovaries. So here's ovary, fallopian tube, and then uterus. This darker area is the endometrium. My source for this, it's work in the public domain, so it was produced by the U.S. government. And then this image I got from Creative Commons license off of Wikimedia Commons. So here we have the endometrium, and actually I want to change the color of that if I can. I'll just do this. So we have the endometrium right here, and then the myometrium, which is contractile. And this endometrium is what's growing and sloughing off in menstruation every month. This is where the bleeding cycle comes from. So the thing I want to point out here is we have this basalis layer and this functionalis layer. The basalis layer is like the reserve cells. These are the cells that have to stay here. If these get damaged, you don't get any endometrium proliferation the following month. It's just done. So these basalis layers so they get acted upon, and then they start reproducing mitosis, and you get this functionalis layer produced out of that. Now there are many actual subdivisions of the functionalis layer, probably not important for this discussion. So estrogen is what's acting on the basalis layer to cause proliferation. So estrogen causes it to grow, and then progesterone causes it to maintain where it's at and cause secretions to come out. If there is no implantation, no rescue of the corpus luteum by HCG, then you have a couple of things that happen. First of all, you're going to get prostaglandins and cytokines and various other protein factors. These do two things. They cause vasoconstriction, and vasospasm. This causes necrosis. So it's not apoptosis this time, it's necrosis. Causes all of those cells to die of the functionalis layer. Then the next thing you get is the myometrial contractions. This assists in expelling all of the necrotic tissue, and that is menses. Implantation can occur on cycles days between 20 and 24 roughly. So what happens is that after ovulation, you have somewhere between a 24 to 72 hour window, depending on what source you read. There's a window for fertilization. And if the oocyte gets fertilized in that time, then it will be able to develop enough that it can implant, by the time it reaches the uterus. So fertilization is going to happen in the fallopian tubes. And then the zygote, the proembryocyte, will begin developing and producing all the different things that it needs, for example the trophoblast, so that by the time it gets into the uterus, it will have time to implant. If it doesn't get fertilized in this time frame, let's say it gets fertilized when it's in the uterus, it's not going to be able to implant. It's going to miss its window for implantation. Now the very last thing I'm going to say is there's a concept called endometrial dating, and basically this is the histologic appearance. histologic appearance of the endometrium. So what happens is somebody took a bunch of normal, or you can say average women, and dated and took samples of their endometrium throughout their monthly cycle and used that to say, okay, when the endometrium looks like this, it should be cycle day 20, or when it looks like this, it should be cycle day 14 or cycle day 25. And so there's this dating method. By looking at the endometrium, you can tell what date in the cycle you should be in. Anyone who does not match up from their actual cycle date with their endometrium, this is called a luteal defect. So luteal defect is basically that you have an endometrium whose actual apparent histologic age is not the same as the cycle age. And if it's more than 2 days, you're going to have problems with conception. You're going to have problems with implantation. And whenever an implantation does occur, you will often have early loss of the embryo, spontaneous abortion. All right, that wraps up the monthly cycle. There's also more reproductive endocrinology as far as the hormones of pregnancy, but I'm not going to go into that in these videos.